Hello students, today we'll be continuing in the second chapter, sexual reproduction in flowering plants. So all flowering plants show sexual reproduction. Flowers are the sites of sexual reproduction. So we'll see how sexual reproduction takes place in flowers. Okay, several hormonal and structural changes result in differentiation and development of floral primordium. Inflorescence bear the floral buds and then the flowers. In your first year class, you might have studied what is inflorescence. You have studied, right? So inflorescence, it's like a group. So in an inflorescence, they're in a group, so that one flower together forms an inflorescence. That inflorescence, they bear the floral buds. First comes the buds, then the flowers. Before studying how sexual reproduction takes place in flower, let's see the parts. Typically, the flower has the two parts, that is androecium and gynoecium. So androecium is what? It's nothing but the male reproductive part of the flower. Gynoecium is the female reproductive part of the flower. So today let us study about the androecium. Okay, it's a male reproductive part of the flower. It consists of whorl of stamens. Their number and length are variable in different species. There they have marked the separate picture of the stamen. You can see them. Means there are many stamens in one flower, and their number and length, they will be varying for different flowers. Next, stamen is having the filament. Observe there, in this stamen picture, there they have given the filament region, and next the anther region. One by one observe properly, children. Only then you can understand the sexual reproduction flowers. Androecium, as I told you, female, no? So mainly there are two parts, filament and anther. In filament, it is long. You can see that picture, that it's long and it is somewhat bent stalk, and its proximus, its bottom portion, it is attached to the thalamus or to the petal of the flower. This anther, its terminal or typically bilobed. You can observe there at the top, bilobed means two lobes, and each lobe is has two theca. In the right, you can see the portion, the children. There is the anther is been cut horizontally. So often a longitudinal groove runs lengthwise, separating the theca. So between them there is a groove is running between the two theca. Now next you'll be studying about this pollen grains. This pollen grains, it is present in this anther. So when they are cut and they are dissected, there you will be observing this pollen grains and the pollen sacs. See, transverse section of the anther. The anther is tetragonal structure, consists of 4 microsporangia located at the corners, 2 in each lobe. It means there are 4 microsporangia region are present, and these microsporangia, they develop into pollen sacs. And there they have marked pollen sacs in the picture. They extend longitudinally all through the length of the anther and are packed with the pollen grains. There will be 4 microsporangia. No, what happens? The top one and the bottom one, they join together. So they will extend longitudinally through their length, and they will be because of the help of this pollen sacs, these pollen grains will be packed in them. So structure of microsporangia. It has a circular outline. It has a 4 layers: epidermis, endothelium, middle layers. Next is the tapetum. The first 3 layers, what it does, it gives a protection and help the dehiscence of the anther to release the pollen. It means the pollen grains, whatever is present, it might burst out and go out. So these 3 layers, what it does, it will be protecting this pollen grains. So, what does the tapetum does? It will help in the nourishment of the developing grains. The remaining 3 layers it will be protecting, whereas tapetum it will be nourishing. And the cells of the tapetum, they will be containing the thick cytoplasm, and generally they have more than one nucleus. In young anther, each microsporangium has sporogenous tissue at the center. It consists of compactly arranged homogeneous diploid cells. Diploid in the sense 2 cells are present, and they are present at the center of the microsporangium. That is when they are young in anther, not when they are matured, when they are young. Now pollen grains has to be produced. So before pollen grains starts producing out, there is a process called microsporogenesis. As the anther develops, each sporogenous cells undergoes meiotic divisions to form microspore tetrads. Meiosis means what? You will be getting 4 cells now. So see there, one pollen mother cell, it undergoes meiotic division and will be getting tetrad of pollen. Tetrad in the sense 4, okay. And it separates to the 4 pollen grains. The formation of microspores from a pollen mother cell through meiosis is called microsporogenesis. So from pollen mother cell, it undergoes meiotic division and will be getting 4 cells. So this process will be called as microsporogenesis. As the anther mature and dehydrate, the microspore dissociate from each other and develop into pollen grains. When the anther mature, no, and they'll get dehydrated. In the sense, the water, the moist condition won't be there, no. During that time, these pollen grains will burst out. Each microsporangium, they contain thousands of pollen grains. They are released with the dehiscence of anther. When this anther cannot hold these pollen grains out, no, they will burst out. These pollen grains move out freely. So there will be many pollen grains. It's thousands of pollen grains. Now let us see the structure of the pollen grain. It is spherical and 25 to 50 mm in diameter, and the cytoplasm, it is surrounded by the plasma membrane. A pollen grain has 2 layered wall, that is exine and intine. Over 60% of angiosperms shed their pollen grains at 2-celled stage. Okay, this first let us see what is this vegetative cell and generative cell. Okay, then we will come to this. Okay, intine, it is the inner wall. Intine, internally, inside, remember in that way. So that is the inner wall, and it is thin and continuous layer made up of cellulose and pectin. And in this matured pollen grains, you are having the 2 cells, vegetative cell and the generative cell. they have marked vegetative cell and generative cell. So compared to generative cell, vegetative cell is bigger, right? Hence vegetative cell, it is bigger and it is having abundant food reserve and large irregularly shaped nucleus. It means this vegetative cell, it will be storing reserve food materials, and the nucleus is not in a shape, it is irregular. Whereas generative cell, it is very small and it will be floating inside the cytoplasm of the vegetative cell. It is not independent generative cell, it will be floating inside this vegetative cell, and it is spindle shaped with dense cytoplasm and nucleus. Over 60% angiosperms shed their pollen grains at two-celled stage. When the pollen grains develop into two-celled stage, no? Two-celled in the sense vegetative cell and generative cell. During that time, this pollen grains will burst out. And in others, generative cell, they divide mitotically. The only vegetative cell, they will divide mitotically, and you will be getting two male gametes, and thus the pollen grains are shed at the three-celled stage. Pollen grain is also called as male gametophyte. Sometimes they will ask you to write about male gametophyte or like pollen grain in that way. They shed pollen grains have to land on the stigma before they lose viability. Means before they lose their property, means before they lose their power. So in that way, they have to land on the suitable surface. So the viability period for pollen grains is variable. It varies for different species and mainly depends on the temperature and humidity. In cereals like rice and wheat, the viability is 30 minutes, whereas in some other members, it is for months, like members like Leguminosae, Rosaceae and Solanaceae, they have viability for months. And economic importance of the pollen grains, they are rich in nutrients. Pollen tablets are used as food supplements. Pollen tablets and syrups increase performance of athletes and race horses. They are stored for years in liquid nitrogen. They are used as pollen banks in crop breeding programs. These pollen grains, though they are very rich in nutrients, they will be used by these athletes and race horses as the syrups and tablets and all, because it's a very good food supplement. No, instead of taking some heavy food, what they does, they will take these tablets, so that they will get enough number of proteins, rich in proteins. So, and this can be stored for years together, means these pollen grains. Hence they are used as pollen banks in crop breeding programs. So when they are stored in these pollen banks, what happens, either they can develop the new species or something, they will find out something new from the pollen grains. A pollen grain of one plant will be injected into other plant and there will be development of new species. In that way also it will be used. And pollen grains of some plants like Parthenium and carrot grass are allergic for some people. It leads to chronic respiratory disorders. It's asthma, bronchitis, etc. Now we don't find these Parthenium plants much. Okay, when we were young, there were more Parthenium plants and it was highly allergic. So the pollen grains of the Parthenium or carrot grass, it's not good. Now what we came to know mainly is these pollen grains, very few pollen grains are not so good and other pollen grains are having much importance. These are the important questions. What are the sites of sexual reproduction in plants? Means what are the main site which is involved in sexual reproduction of plants, and which are the two parts involved in sexual reproduction of plant. The main two parts. Explain the structure of anther. You have to explain as well as draw the diagram, that is microsporangium diagram. What is microsporogenesis? This is 1 mark. Draw neat label diagram of pollen grain and explain. You have to draw the diagram as well as you have to explain it. It's for 5 marks. Third question, fifth question is for 5 marks and it is important. In a practical point of view also, these two questions are important. That is third and fifth one. Thank you.